Welcome to the second lecture on research design and data management. We are going to today be talking about experimental design. The first lecture, we figured out how to ask a question and what we might ask a question about, and then how to frame that question scientifically and write an hypothesis. This lecture, we are going to be talking about how to design an experiment to try to falsify that hypothesis, which is following the scientific method. All right, starting at the very beginning with designing an experiment, uh, you have to conceptualize it and think about it and try to figure out what's realistic and what's not. And also, you have to make sure that it actually answers your hypothesis, your research question. For example, you may really love Teslas and want to do experiments on batteries at the Tesla plant, but you don't know Elon Musk, and you probably aren't going to be able to get access to that unless you're very lucky. So you have to figure out what's realistic within your budget, within with the equipment that you have, and the time that you have, most importantly, and then you can figure out whether that is a realistic plan or not. It's really important to set things up well before you get started. So here I want you to stop the video, and we wrote down some individual research questions from yesterday's lecture, and you came up with some questions that interested you. And so I want you to try to conceptualize an experiment that would answer the hypotheses that you wrote. What would we need to do it? How much does it cost? When would you do it? And how long do you have? Take a few minutes and write down, at least for one of them, what your experimental design might look like. And then we can think about that, and it'll be much more realistic and personal, and make a lot more sense for you as we go through this video, as you compare it. Okay, did you stop the video? No? Okay, go back. Go ahead and stop it now. All right, if you did stop the video and wrote out your example, then you're going to have a lot more. You're going to get a lot more out of this this lecture. But for now, we're going to use a question about penguin growth rates. So the example, I like penguins, so I would like to look at emperor penguin growth rates. And how would we go about doing this? Same questions. What would we need? How much does it cost? When, in terms of time? And how long do you have to do it? Well, we could do this experiment in lots of different ways. And if we want to look at growth rates in the wild, we could go to where penguins live, which is Antarctica. And we'd have to get a plane ticket there and back. We need to find some penguins. We need lots of cold weather gear. We need to rent some space at the Scott base and or some other Antarctic field station. So that sounds like it'd be pretty expensive to go down and do all of that. But the other option, if we're going to look at penguin growth rates, is to do it in some sort of controlled environment or lab. And how much would that cost? Well, we've got to think about building some place that would house them and give them the environment that they're suited for, because you want happy penguins. So you need to have refrigeration. It's got to be cold. They're used to snow and ice under their feet, so they like that kind of thing. That's what you put there. You're going to have to have all the equipment to look after them and clean the water that they're going to be swimming around in at their little artificial beach. And people that are going to look after them, and that's salaries, food, all of that kind of thing. And so it may sound like option one is expensive, and it is, but option two is likely to be a lot more expensive. So these are the kind of things that you want to think about when you're conceptualizing your experiment. And when would you do it? Most people work. Can you take 3 months off to go to Antarctica? Maybe you can, maybe not. What about what time of day will you weigh them? Will you weigh them all at noon? Are you going to weigh them first thing in the morning when they've been overnight in the cold and they've been shivering all night before they've gone out to forage? Or are you going to weigh them all during the evening, or are you going to think about what time you're going to weigh them? Is it going to be in the middle of the summer? Are they fattening up for, if you're using weight as a proxy for penguin growth, are they fattening up for their winter? Well, you wouldn't call it hibernation, but when they just stand there and use up all their fat stores. Is that the right time of year? Are you going to be able to do that year after year to know how much they grow? How long is it going to take to measure them? If you've only got 2 weeks down there, are you going to see much growth rate in 2 weeks? How about different life stages? So do they grow very quickly as young ones and slower as adults? Are you going to just measure the young ones or just the adults? These are all different kinds of questions that need to be thought about when you're designing your research experiment. Okay, you got your plane ticket to Antarctica. You're going to go measure some penguins. You know when you're going to do it. You know how much money you've got in order to put towards it. So next question. What type of study? Let's get into the nuts and bolts of designing an experiment. First little bit of terminology for you here. Experimental versus observational studies. So think about an experimental study as you're doing something to somebody or someone or something. You're making it do something, or you're subjecting it to something. So if you're going to just observe what happens without doing something to something, then you are doing an observational study. All right, so if you want to, for example, check what happens to lung capacity after people smoke, then you could do that experiment with, especially in women. You can do that experiment. Take a second, think if you how you would design an observational study and an experimental study. There's no right or wrong answer. Just give it a go, and then once you've done that, start the vid again. Okay, hopefully you've come up with a couple of studies, and this is what I came up with. Check it against yours and see what you think. So an experimental study, you are going to find 100 women, if that's what your subject size or your sample size wants to be, 100 women. You want them to all be about the same age because lung capacity will vary with age. And you get 100 women that don't smoke. And so 50 of them, you say, here's 10 smokes a day, half a pack a day, or maybe even a pack a day. And you say, you need to smoke all of these every day, and we're going to make you smoke for 10 years. And then we're going to see what your lung capacity is. And then 50 women, we're going to say, you can't smoke for 10 years, and then we're going to measure your lung capacity. So that's assigning a treatment. You are experimenting with them. You are making them do something that relates to the treatment. So if you're going to do an observational experiment, you just go out and find 100 women, and you find enough women that are age 30 that you have you take the first 50 that do smoke, and then the first 50 that don't smoke, and there's your sample. And you're just observing what happens, what they've done to themselves. I mentioned in the last slide a couple of terms that we should probably define. The first one is a treatment. So this is whatever the thing is that you are interested in studying. It's a thing that is potentially having an effect. So in the women's smoking and lung capacity, we're wondering whether smoking affects lung capacity. So smoking is the treatment, and we will have a treatment and a non-smoking treatment group. One to which we apply the treatment to, and one not. The experimental unit is those individuals within the group that you do apply the treatment to. So every woman who you made smoke or who already smoked and you observed, that is, she's one experimental unit. And then we have the control, which are the experimental units or individuals. Those are the women who didn't smoke and no treatment is applied to them. And so if the only thing between the experimental units, the control and the non-control groups, is that experiment, that thing that you applied, then that means that any differences you see at the end can be put down to the treatment. You might not always have a control group though, and sometimes you need a reference site. Let's say you want to look at the effects of some sort of effect of pasture compared to native land. Now, you're not going to take 2 blocks of native forest and cut down one of them in order to have a new paddock to study. You're just going to use an existing paddock, and then you're going to choose a bit of native forest that is similar in terms of terrain, in terms of steepness and gradient and rainfall and everything else that you can possibly think of that's similar, and use that for comparison. So that is a reference site. I should add to that last slide that a reference site, finding a good reference site that we haven't done something to, is usually pretty difficult in ecological studies. For you guys who are going to be studying ecology, it's pretty tough to find something that we haven't altered in some way. And that leads to the fact that observational studies don't have as much statistical power. We can find correlations, but because we haven't manipulated the 2 groups, and the only thing that's different between them is the treatment that we've applied to the treatment and control groups in an experimental type of study, the observational studies give you correlation, which is not causation. So you need a lot of observations in order to get a good, strong inference from an observational study. So we're going to Antarctica. We've decided on whether we're going to do an observational or experimental design to the study. And now we've got to talk about what variables we're going to measure. Okay, in the interest of this video not being 700 minutes long, you can pause this and read through these. Essentially with variables, that's the stuff that we're going to measure. It's a factor, a trait, condition. They can be qualitative, quantitative, derived, independent, dependent, and controlled. And we're going to go through and talk about what those variables definitions are. Starting with an independent variable. This is the one you are trying to control. It's usually in an experimental design, it's the treatment. So for example, if we're going to do an experimental design, we're going to control the food that we are giving penguins and see what their growth is like. We'll give them silverfish, squid, or krill, and that's all those individual groups get to eat. And we're going to measure how fast they grow. That is the independent variable. In the experimental design, we manipulated their diet, and that was the independent variable, the type of diet. In the observational study, we go and find something that may be different about that population and look at that thing and see if we think that that's having an effect on, for example, penguin growth. And we'd look at these different areas and look at the populations in those different areas and see if there was a difference in the penguin growth. So again, that is the thing that we're postulating affects something else. That's the independent variable. And this one might make a little more intuitive sense. The dependent variable depends on the independent variable. So in this case, the independent variable was the food type, and what we measure is the growth. The growth depends on the food type, which is the independent variable. The dependent variable growth depends on the independent variable food type. So it's the thing that we are interested in finding out if there's an effect from the treatment. In this case, the treatment is food, and we're wondering if there is an effect on penguin growth. That is the dependent variable. And for our observational studies, it might be the same thing. It's the variable that depends on the independent variable. Okay, so we had these different sampling locations, and we called these the independent variables, or it wasn't a treatment, but it's the thing that we're varying between the populations. But we still want to find out if penguin growth depends on the independent variable of location. And finally, the controlled variable, that's something that we don't change between groups. We're hoping it's exactly the same so that we can say the effect of the independent variable is the strong one that we're measuring. It's the important one. So for example, if you're giving food to the penguins, you give them each the same amount. You might give them 500 grams each per day, no matter what type of food you're giving them. And you're using the same species of penguin, and you're measuring the penguin growth through the same period of time, through the same season, and you're using the same observers and the same scales and the like, so that you can say that the only thing that really varied was the control variable, or the independent variable, the treatment. Observational studies, same thing. Like the prey population, you're looking at, you're trying to maybe keep the penguin, you're looking at penguin colonies that are all very close to big krill production areas. So you're trying to say that the location matters in terms of growth, but most of the other factors are going to be the same. In observational studies, it's very hard to control for everything, and you're going to do your best, but that's why these things have less statistical power. There's a lot of stuff that can get in the way and confound your results. So that'll be the end of this video. We're going to go on to a little bit more of experimental design in the next. Otherwise, this thing will be too long. But you have practice to cement those definitions in your mind on variables in the workbook, page 19. Thank you.